All right, so we left off last time talking about transcription, where DNA is used to make messenger RNA, right? So that messenger RNA then has to be translated into protein, okay? So during the process of translation, you get a functional polypeptide, and it's going to be made using the information in the sequence of codons in the mRNA strand, messenger RNA. All right, so that sequence of codons is then determining the sequence of amino acids. So translation then is going to proceed in 3 steps. Okay. All right, so you've got initiation, elongation, and termination. So in initiation, the messenger RNA is going to attach to the ribosome. This is after it's already left the nucleus, gone through a nuclear pore. Okay. So it's gone to the ribosome. And then transfer RNA, tRNA, is going to bring the corresponding amino acid to the messenger RNA. Now, the transfer RNA has an anticodon, okay? And it's going to match the codon, or it's complementary to the codon on the messenger RNA, okay? Then we get to elongation. And here, another transfer RNA is going to bring another amino acid. All right, and it's going to bind to the next codon on the mRNA strand by complementary base pair rules. Okay, so you basically have 2 sites filled, right? 2 codons. Then you're going to form a peptide bond between the 2 amino acids. So there's going to be a chemical reaction that's going to link the 2 together. All right, link the 2 amino acids together. And then the first transfer RNA can leave at that point. Okay, so all right, so then it's going to bind. So a third transfer RNA is going to come by and bring another amino acid. And then it's going to bind to your next codon on the messenger RNA strand. Okay. All right. Now in termination, elongation is going to continue until you reach the stop codon. Okay, there's one of those codons that codes to stop protein synthesis. And it's going to cause the ribosomal subunits to detach from the messenger RNA. And then you may have in some cases, you get a polyribosome, where there's a series of ribosomes attached to the same RNA strand. Okay, so this is like, let's just say there's a sequence that you got to make a whole lot of protein from. So basically, the ribosomes will get in a line and go down the mRNA, and one will follow on behind it. And you just end up making lots and lots of protein. Okay, so that is translation. So what I'd like for y'all to do is talk to your battle buddy and explain the steps of translation. So one person can explain, and the other person explains, and you can do this at home as well. All right, just talk a little more about DNA and the control it has over cell structure and function. I mean, we've talked about that specifically with transcription and translation, but let's talk about it a little more generally. So how does DNA control the cell? Well, I'm glad you asked. So it controls the cell by directing creation or production of proteins. Okay, synthesis is another word that means making or proteins, or making, synthesis, production. I mean, that's what synthesis is. So DNA is going to have direct control over the synthesis of structural proteins in particular. So like your cytoskeleton, your proteins that are in your membrane. All right. DNA has indirect control over basically all the other parts of cellular metabolism. Okay. And it does that by regulating production of enzymes. Remember, enzymes are a type of protein, right? And they catalyze chemical reactions. So they don't necessarily directly control the other organelles, but they control the enzymes that work in the organelles. Does that make sense? Okay. All right, so Cell life cycle. So a cell cycle basically is a period from the beginning of one cell division to the next cell division, okay? And what is cell division? Well, it's reproduction of a cell where one cell divides and becomes two cells, makes two daughter cells. And why do cells divide? Well, they increase the size of an organism, right? More cells typically is going to mean more size. And they can replace cells that are dead or worn out. Okay, cells get beat up, worn out, just like, you know, anything else. All right, so again, DNA, remember deoxyribonucleic acid, that's our genetic material in our living organisms. All right, it's our blueprint for, you know, who we are. All right, and what are chromosomes? So chromosomes are structures inside your nucleus, and they contain the genes for what we call genetic expression, right? So when we say expression, what we're talking about is making protein, okay? So the process of transcription and translation is basically how the DNA expresses itself, okay? So humans have 46 chromosomes. Other organisms, some have more, some have less. We have 46. And what are genes? Well, genes are segments or units of DNA, of genetic material, that are localized in a specific spot on a chromosome. And they determine some sort of inherited hereditary trait, right? The chromatids are, or a chromatid is one copy of the two copies you have in a chromosome. It's surrounded by a protein complex that we call a kinetochore, all right? And so it's like DNA wrapped around some protein, okay? Now the chromosome is actually composed of two chromatids, okay? So, and they are joined together at what we call the centrosome. And the centrosome is the place in the middle. So a chromosome basically forms an X. And the lines are the chromatids. And then the little space in the middle, that's your centrosome that holds the chromatids together. All right, now, we also have centromeres that we have to be concerned with. And that is a space, excuse me, and I want to make sure I say this correctly. The centromere, rather, excuse me, is where the chromosomes attached to. So let me find a pen. So all right, so let me make sure I'm saying that correctly for you. All right, so let me switch to show you what I just wrote on the board. Do a new share. All right. So trying to get out of the shot here. So the whole thing is a chromosome. The whole thing there is a chromosome. All right. So like from here down like that, like, kind of like a less than or greater than sign, that's a chromatid. Okay. Now holding two chromatids together is a centromere, or is a centromere, yes. So that's, just want to make sure you understand. Chromatid, two chromatids make a chromosome. They're held together by a centromere. Okay. So just want to make sure you see that. Switch to share. All right. So the mitosis then is nuclear division. So you're dividing the nucleus up, the genetic material. So you end up with two nuclei. Same genetic material as what you started with in the original. Both, they have two copies essentially with the same information. All right, and it happens in somatic cells. Remember we talked about two different kinds of cells. We talked about sex cells or germ cells and somatic cells. So mitosis happens in somatic cells. Okay, it's going to preserve DNA from one generation to the next. You have meiosis, which happens in sex cells. Okay, it's just a form of nuclear division in sex cells, and you actually reduce the chromosome or the chromosome number by 50%. All right, so what you end up is with cells that have 23 chromosomes instead of 46. Okay, and then also, typically with a different number of cells, right? So we'll get into more of that later. All right, so cytokinesis means you're dividing the cytoplasm, and this typically happens during telophase of the cell cycle. Okay. All right, so let's pause here and talk to your partner about 3 things you've learned about the cell cycle. You can do this at home as well. All right, so a little more about mitosis specifically. So, or the cell, actually the whole cell life cycle. So before we even get to mitosis, let's just talk about the steps in the cell cycle. So interphase is when most of the time your cell is alive, it is in interphase, right? Or most of the cell life cycle is in interphase. And it has some phases within it, G0, G1, S, and G2. So in G0 or zero gap phase, your cell's just doing its normal everyday business, not preparing for cell division, okay? So once you get to G1 or first gap phase, then you start preparing for cell division. All right, so you start making copies of the cytoplasmic components, things in the cytoplasm like your organelles, all right, membrane, so you're trying to double up so you have enough for 2 cells in G1. Then we get to S phase, and S stands for synthesis. Remember we said synthesis means making, right? So here we're copying or making DNA. This is where DNA replication happens, okay? So, all right, so DNA replication, the 2 strands come apart, and so this is what's different about replication versus translation or transcription. In transcription, one strand is used, one is the coding, one's the template. In replication, they both get copied because you're making 2 copies, okay? So, all right, and that way each daughter cell has its own complete set of DNA. All right, then you get a second gap phase. Okay, and that's when you start replicating your protein or synthesizing your proteins rather, excuse me. So that's where your transcription and translation happen, okay? Now, that's not to say you don't have any of that going on at other points in the cell, but you basically are having a lot of protein synthesis going on because you're gonna have to make enough for 2 cells during G2. All right, then we get to M phase. Okay, and that's where mitosis occurs. All right, so mitosis has its own phases. All right, first phase is prophase. So here the DNA is condensing, and when you say condensing, it means it's packing tightly together, right? And it's forming the chromosomes because they were basically chromatin during interphase. All right, so it's a little more spread out. Then it packs together into chromosomes in prophase. All right, then you start seeing spindle fibers, right? They start attaching to your chromosomes at the centromere, okay, that middle point, right? Centromere. Nuclear membrane starts going away, okay? So at the beginning of prophase, you can see the nuclear membrane. At the end, you cannot, okay? All right, then we get to metaphase. And the M for metaphase, once you remember, stands for middle, all right? So chromosomes align along the middle of the cell. All right, so you form a straight line. So metaphase, all right, we're not going to draw all 46 chromosomes, but essentially cells are going to be lined up in a line, or chromosomes will be lined up in a line in the middle, okay? All right, those spindle fibers. You can see those are attached there at the centromeres. We call this the metaphase plate, a line, all right? Line in the middle, we call it metaphase plate. All right, then we get to anaphase. And in anaphase, those chromatids or sister chromatids split apart, okay? And we start seeing those migrating to the different sides of the cell. They're being pulled by those spindle fibers. And on these ends, we have centrioles over here, okay? On the ends, on the poles of the cell. All right, so they're also attached to those spindle fibers and they pull the chromatids apart, okay? But if you see them split apart, you're in anaphase. All right, then we get to telophase, and telophase is kind of like the reverse of prophase. All right, so your chromosomes are at the poles or the sides of the cell. All right, remember we said cytokinesis tends to happen at the same time, so that's when your cytoplasm starts dividing. All right, your nuclear membrane starts to reform. Okay, so you start seeing it again. Nucleolus starts to reform, you can see it again, okay? Chromosomes start to basically, they're not condensed, right? So they're harder to see. The DNA is more spread out within the nucleus. And then on the edge of the cell membrane, you get a cleavage furrow, okay? So, and because again, all right, this is the cleavage furrow, okay? So now you've got your DNA is no longer in chromosome form, and you have the DNA that's spread out in the nucleus, all right? So you've got different, basically you can see the line where the new cells are gonna form, and then eventually you end up with just 2 cells, all right? With the nucleus. So that's your telophase, prophase, metaphase, anaphase, telophase. And you end up with 2 identical cells, all right? So, all right, so let's talk about mitotic rates. So just a rate is how fast something happens, right? So it's the number of cells in mitosis at any given time. It's used to estimate the frequency of cell division. So in general, longer the life expectancy of a cell type, slower the mitotic rate's going to be. Okay, if it lives a long time, it doesn't have to divide as fast. All right, if it doesn't live a long time, it divides quickly. And stem cells, these are cells that can differentiate into other tissues, right? So they are going to undergo frequent mitosis, right? So get replaced, and they're going to replace more specialized cells, right? So they're differentiating. All right, so how's their cell cycle regulated? Well, it's through genetics, your genes. So you've got growth factors. These are compounds outside your cell. They're extracellular, and they can stimulate cell division. They can stimulate growth. All right, so some of that stimulation is good, some of that stimulation is bad. All right, typically tumors are bad. All right, another word for tumor is a neoplasm. It's just a mass or swelling that gets produced by abnormal cell growth or cell division. Okay, tumors can be benign or malignant. So if a tumor is benign, it just tends to be there, but it doesn't threaten the person's life. And it just typically remains in what we call connective tissue capsules, kind of stays where it is. Now, typically when a tissue is malignant, it spreads to other tissues. We call that metastasis. It metastasizes and it goes to other places. And it is usually life-threatening, okay? You try to catch tumors before they become malignant, right? So, and we're talking about this because this relates to the cell cycle, right? So cancer is basically the cell cycle gone wrong, right? So the invasion here is a process when the cancer cells start spreading to surrounding tissues. We call that, and it's invading those tissues. Again, metastasis is when those cancer cells disperse to more distant organs and tissues, okay? And they can set up secondary tumors there, right? So that's why you want to try to catch cancers before they become malignant, right? Because sometimes you'll catch them, it's too late and they've already spread somewhere else, even though you cut the cancer out where it was, right? And cancer is that illness that is caused by these malignant cells. Okay. So I want to end with just about the concept of differentiation. So you've got all kinds of different cells in your body, right? I think there's over 200 different kinds of cells. And it's specializations due to regulation of your genes and your DNA. It produces cells that have limited capabilities. They're all, all your cells don't do the same thing, right? Some do one thing, some do another. All right, so basically these specialized cells in certain areas are going to form tissues, right? And each tissue is going to have its own functional roles. So we've already started talking about tissues in lab, and we're going to start talking about tissues in lecture. Okay. So that is chapter 3. All right, so your test is over, chapter 1, 2, and 3, right? So what I do want to tell you is, I've got to grade your essays and your short answer, your fill in the blank, all that manually. So when you take the test, Canvas is going to try to grade that. Okay, so if you don't type exactly the way it has in Canvas, it's going to count it wrong. Now on the essay, it's just not going to grade it because I have to grade that. But for the short answer and the multiple choice or short answer and that kind of stuff, fill in the blank. If you don't type it exactly the way that it is in Canvas, it counts it wrong. I have to go back in and regrade it. Okay, so don't email me and say, "Hey, I got this right and Canvas said it's wrong." Like, I'll work on it. Okay, like I'll get to it because I know that already. I know it does that. All right, so also just don't leave any questions blank. Answer them all. Don't hit submit before you do them all. Okay, because if it's blank, it's wrong. All right. If it's multiple choice and you put something down, you have a chance of getting it right. Fill in the blank. If it's blank, it's wrong. All right. Under no circumstances do I want to see a test turned in with no essay. Okay. I'm not looking for you to be the next Pulitzer Prize winner, okay? I just need you to answer a few sentences or answer a question. So under no circumstances do I want to see a blank in that essay box. Are we clear on that? Okay. If we go back to the modules and go to start here at the bottom, it says you the essay grading rubric for the exams. Okay, so we'll go ahead and make this bigger so you can see it. So it tells you here, completion, comprehension, organization, grammar, and spelling. Okay, so this is how you can get full points. Now, sometimes the essays are worth 12, sometimes they're worth 20. So it's just what I have to do is I have to scale it. So whatever you get out of 20, I scale it to however many total points that test is, how many points the essay is worth on that test. All right, so, but if you have an answer that's complete, including all pertinent information, you can get a 5 for completion. If your answer indicates a deep understanding, you can get a 5 for that. If your content is extremely well organized, logical, and easy to follow, you can get a 5 for organization. If you have no major grammatical and spelling errors and it's easy to read, you can get a 5 for grammar and spelling. Then you can get a 20. Or you can get however many total points that question is worth on that exam. Okay. It is not uncommon for students to get full credit. Okay. So, and you don't have to be the best English writer. You just have to be able to follow directions. Okay. Does that make sense? So if you do that, you can get full credit. Now, you may say, this is A&P, why do I have to write? You are college students and writing is an important skill. Again, I don't have time to read 50,000 pages about why you like A&P. I can read an essay, I can read a paragraph, right? I can read a paragraph. That's what I'm asking you to be able to do. Because honestly, in real life, what do people want you to do? Communicate clearly and concisely, right? So you just need to be able to tell me clearly and concisely how to answer the questions that are on that review sheet. And I gave you the questions, right? It's not like you don't know what questions you need to do well on. My best advice to you is get on a computer or get on Google Docs or whatever you use to write on, type your answers out, practice. You won't be able to use them on the test, but you just get yourself used to writing them out, okay? So also quiz yourself, okay? You can do well on this. And also just, I don't know what kind of grades you're used to making. You may or may not make the grades you want to make on this first test, and that's okay. You can still make the grades you want to make on your final grade. I want to make sure you're clear about that. You have a lot of assignments. It all averages itself out, okay? So don't think if you don't get the grade you were wanting to get on this, that it's over. It's not by a long shot. And the people who bail on the first one are always upset that they bailed because everybody else who stuck it out got what they wanted to get. Okay, so don't be that student. All right, just stick it out. You'll be fine. All right, I've had plenty of students get A's. I've had plenty of students get B's in this class. All right, and some of them were always A and B students. Some of them were not, and they became A and B students. Okay, so regardless of which one of those is you can do this. Okay, so is this class harder than probably some of the classes you take? Okay, so raise your hand if you think this class is harder than some of the other classes you're taking. Okay, look around the room. You're not alone. Raise your hand if this class requires more study time and more assignments than other classes. Look around the room. Okay, it's not just you. Okay, so don't feel like you're in a, you're struggling and nobody else is struggling. Okay, you're all struggling together. Okay, so it's gonna be all right. So do your best. It is on auto lock, so no resources. But I really try to tell you on the review sheet what to study, so you know exactly what to study, and I try to make it so that I'm not looking for a random fact on page whatever, and what it is you really, really, really need to know out of this class. Okay. you know, because I don't like you not knowing what to study. So, all right, so we good there as far as knowing what to do to be successful on this first test, okay? So we'll probably go over it the week after. Give me a few days to grade it, all right? So with that, all right, I think we're gonna go ahead and start chapter 4. All right, so I'm not gonna end the meeting because we got y'all gonna watch, but I'm going to